The adenovirus has a linear, but the prokaryote the prokaryote is a linear?
Circular.
It has a desoxy-DNA genome, early region and late region, you understand it. Complex transcriptional unit, single promoter it has, but the bacterium prokaryote has a lot of promoter. It can be start anywhere of the circular DNA. We have an alternative splicing. What does it mean, alternative splicing?
Oh.
It's here. For example, you want to understand what does mean alternative splicing, okay? See, these are the different...
I remember. Like if you have like 1, 2, 3, the 3 can come like after 1, right? For instance.
I don't understand. What do you say?
Like here you have like, okay, 1, 2, 3, right?
Yes.
Alternative spacing could be like 1, and then maybe after 1 can come 3.
Yes, it
Right?
means for example, maybe we cut all of them.
Yeah.
We cut this
But
one.
the
We cut this
combination
one.
of them is like different.
It means
you
we
know?
have a different mRNA,
Yeah.
and every of them do a different thing in different organ.
Yeah.
They have a different job. It means it's an alternative splicing. Maybe you see it cut no intron. Maybe it cut some of them, all of them, more than all. You see that this is shorter. When it's shorter, it means it cut a lot.
Oh, you can cut it more. Is that what it is?
For example, I would like to show you. This is the mRNA. Intron. Good.
Yeah.
First of all, I just remove one of the intron. 2 of them is remain. Then I remove the other 2, and I remove all of them. I have a 3 mRNA.
Okay, that's alternative.
This is the alternative splicing. 5 alternative polyadenylation element it has. What does it mean, 5 alternative polyadenylation element? It means it can finish the mRNA transcription at the first one, or maybe at the second one, or third, or fourth. It means it will be longer, or much more longer, because the polyadenylated element is good for finish, stop the transcription.
Okay.
Good. Now, what is the exon? Is a leader sequence, the body sequence, and intron it has. But the prokaryote doesn't have intron exon, but the virus has. The virus is like a eukaryote, has an intron exon.
Okay.
Has an intron exon. The virus
But this is for
has, but prokaryote no.
No.
This is a eukaryote, also
And this is-
eukaryotic,
yeah.
and not the virus.
Virus.
Now you learn this one also. Come here, you understand the alternative. Mechanism of splicing, 5' split site, 3'. We need the spliceosome, but spliceosome, it has a pre-mRNA, snRNA, and protein, a lot of U, and Lariat formation.
What does the snRNA do here?
A small nuclear ribonucleic acid.
Okay.
A small nuclear, you don't know this?
I know it, but what does it do? Like.
What does it do? They are the U. U is a snRNP. snRNA 1, snRNA 2, snRNA U1. Okay? The other name. For example, you are Aisha something, but I want to say to you, Aishi. Okay, this is snRNA, but the name is U.
Okay.
Good. This is the other name. Nickname.
But it's snRNA.
snRNA, all of them.
And what does it do?
It comes here and fold the intron like this, fold it, and then when it's folded, it can be cut and go away.
It cuts it. It folds it in the lariat. Formation.
Lariat intron produce.
And why does that happen?
To can be cut.
Okay.
Because I cannot say please remove it, but if it's there and said okay, go away. Understand?
Wait a minute. You cut it first. That's how it goes, right?
No. What's happened exactly? We should understand between 2 exons. We have a GU, R, and IG. AG and GU is a split site, 3' split and 5' split, and the branching point is A. What's happened in the lariat? The lariat has a RGU, RG. Again, RGU, RG. And do you know what it means, RG, in the version?
No.
RG means sister.
Oh.
And U is you.
Oh.
RGAU is your sister. And RG. RGU, RG. Cleavage at 3' split site and splicing of exon happens. RGU, RG. This is important.
G-U-R-G.
Again, this is the RGU. You see the SNRMP1 U1 comes here, bonds to GU. GU has one. The U2 comes bond to branching point. You see?
Once again.
U.
What?
U comes here and bonds to GU.
G.
The U2 comes and bonds to A.
Okay.
We have a U2A. Then other U4, U6, U5 come and bonds to all of them, and then it's exactly a spliceosome assembly. Then the lariat formation happens. Ligation of exon do by the help of U5, and everything is go away. This is an intron lariat.
Ah, okay. I understand. That's why it was so hard to understand, because like so many things are happening, but I get it. This one comes to this, then this one comes to this, and this one comes to this. So the thing is just it separates the lariat formation, the introns from the exons, right?
Yeah.
Yeah, okay. Then I get it. And all of these are snRNAs.
What?
All of these
Yes,
use
all
are-
of them.
Okay, are snRNAs. What about the proteins?
What
Were-
is the difference between them? The amount of nucleotide is different. For example, U1 is so small, but U5 is much more bigger,
Oh, okay.
heavier.
Okay.
No. Abnormality of splicing in the SLE, systemic lupus erythematosus. Do you know what is this? It's an autoimmune disease, abnormal SNRMP, and they lose the hair here or here. This is a very bad autoimmune disease, okay?
And that is because of the abnormal
abnormal splicing.
Okay.
And thalassemia,
Yeah.
and forget the other. Okay?
So I just have to remember.
Yes,
the
you
lupus
should,
and
but-
thalassemia.
Just these two is important. The two name of the
Okay,
disease.
I can remember this.
SLE,
SLE and
thalassemia,
thalassemia.
SLE, thalassemia, SNRMP, abnormality.
Okay.
Like
I
that.
can remember because both of them have L, and it's L in splicing. You know.
Yeah, good.
Okay, I can remember that.
Now, RNA editing, post-transcriptional changing, adding or deleting one nucleotide in mRNA.
And this is now prokaryote, or what is this?
Again, and we want to do the RNA editing. We are always in eukaryote and virus. The prokaryote doesn't have splicing because the prokaryote doesn't
it
have
doesn't have
intron.
it. True.
Post-transcriptional changing, adding or deleting one nucleotide in mRNA. I want to add or I want to delete nucleotide in mRNA.
After you have had
After
this.
it's finished, yes.
After this, you
After.
can add or something. That is called RNA editing.
Yes.
Okay.
Post-transcription, after the transcription. Apolipoprotein B is ApoB100, produced on unedited mRNA, secreted by liver cell into the blood. Component of LDL particle, low density lipid, and ApoB48 is editing cytosine to uracil. Stop codon created, premature termination of protein. What should we remember? Just remember that the ApoB100 apolipoprotein is secreted by liver into blood, is a component of LDL particle. Later you learn
What
if this
exactly-
is a LDL.
Is this an enzyme?
No, this is a protein. This is a
Okay.
lipoprotein added. This is a the name of that was
What was it? Protein, lipid, what?
yes, it's a saturation of the cholesterol. The name of that is cholestyramine. Not is I remember. I forget it. Sorry. It has a lot of cholesterol. The reach of the cholesterol.
What is this exactly?
You see, the cholesterol is lipid. Yes, the cholesterol cannot come to the blood. All of the cholesterol esterify. Cholesterol is esterify. In this case, they will be hydrophile. Then when they are hydrophile, they can come to the blood. And the ApoB100 comes here. This is the ApoB100. And said, yes, please comes to the blood. Secreted by liver cell, comes to the cholesterol and take them to the blood.
Does it make them hydrophile?
Yes. In this case, if somebody we add ApoB100, all of the lipid will be soluble in the blood and they lose the weight. This is the treatment of obesity.
Okay. What about the apple?
No, just remember this one and see chapter 33.
Okay.
33 here and not here.
You mean topic number 33?
I don't know. It's written here, see chapter 33.
Maybe page.
No.
That's 10. So it's 11.
Chapter 30.
That's 11. So it's 29. 32. 33.
33. This one. LDL building, this one. No, that's not here.
Okay.
I should show it to you. Always when I want to learn something, I should search a lot. Then I imagine I see it face to face.
Yeah.
And that's better to remembering.
Yeah, I know. I do the same thing. I can't do
Probably.
like only like like Sabina. She just read everything, you know, and then I tell her to explain me, and then she only remember a few things, you know. I don't like that because I like to work on one chapter very good and remember it, understand it completely, and then go to the next one. It's just I can't do that.
This is, see, this is a cholesterol,
Yeah.
phospholipid, cholesterol ester, esterify,
Yeah.
and the ApoB100 non-exchangeable come here, and it's secreted by the liver. If somebody has a liver disease, it doesn't produce, and this is a problem to absorb the cholesterol in the body. This is one of the problem. And the ApoB100 said, please, cholesterol comes to the blood, doesn't accumulate in the body.
Okay.
See, and this is a LDL. The name of this is LDL. You see,
Or
it's a
LDL particle.
It's
Yeah,
a LDL. Or VLDL or LDL. We have a ApoB100. We have a lot of ApoBE.
What about this one, Apo? This is. I
No.
don't have to remember it.
No, ApoB48 do something different. The ApoB48 edit the cytosine to uracil in mRNA and stop codon create. When the stop codon is created, for example, what was the stop codon? UAA, UAG, UGR, yes? Now, imagine that we have
to
this
remember this
codon.
one?
You should know the stop codon. You don't know it?
No.
You should learn it before. You should know. If you don't know it, you fail. Because it's the MRT question also. Name the stop codon.
Okay.
URUGR.
Okay.
Again.
I remember in the genetic code, there's this.
64
Yeah.
and 61 is a mRNA. And you know why 61? The codon has a triplase. The codon has a
3 place.
3 nucleotide.
Yeah.
How many nucleotide do we have? 4. It means the probability is to one of the nucleotide is 4.
Yeah.
Is 4.
Uh-huh.
Is 4. Multiple each other is 64. But minus 3. Why? Because this 3 is a codon terminal. It's a stop codon. This 3. It means 61 tRNA we have. 61 tRNA.
I get it.
Now, imagine that this is a nucleotide in the mRNA and I edit the C to U. What's happened? It's, for example, this is a mRNA. It should be a stop here, but I edit CGA to UGA. Okay, it's a stop here. Now I have a shorter mRNA. It means I edit mRNA. Understand? Actually, this is a point mutation sometimes. Point mutation, it means change of one nucleotide to other, and it causes the termination of peptide synthesis. And
Can you write the point mutation?
is a point mutation.
Okay. So that will
But
make
point
this
mutation
transform-
means bad one,
Yeah.
but here sometimes is good one, because I want to learn ApoB488 and see here. RNA editing, synthesis of ApoB100 and ApoB48. This is a DNA. From the DNA, we translate, transcript, and mRNA is produced. Cap formation, poly-A tail. Initiation codon, stop codon. This is a ApoB100. From initiation until stop. This is a CAA. If I change the C to U, in this case, it is a stop codon, and from initiation until this stop codon, I have a much more shorter And the name of that is ApoB48. What is the job of that? The ApoB48, it helps the lipid uptake in small intestine.
Okay.
If somebody has this one, a lot of lipid absorb. The obese people, obese children we have.
Ooh.
Understand?
Okay.
And we should treat this genetic. We should do that. Later you learn also the name of this enzyme that changed this year, and we should inhibit this enzyme. Please don't produce ApoB48.
So the obese people, they have this.
This is a genetic hyperlipidemia.
Oh, okay.
This is a familial hyperlipidemia or genetic one. You learn it later. Just now you should learn the ApoB48 is produced because of the new stop codon formation
Yeah.
in the middle of the ApoB100.
Okay. And it helps with the lipid uptake.
Yes.
Yeah. Okay.
I hate this job.
Okay.
What is this? The role of mRNA, tRNA, and ribosome in the protein synthesis. I think you know this one also. That's very easy.
Yeah, sort of. Yeah.
I want to ask you a question.
No, don't ask me a question.
Name the post-transcription modification of tRNA.
Of tRNA, that's the acetyl, right? That's the acetyl.
This is a tRNA. What's happened here? Which type of post-transcription modification happens here? CCA bonding. Again, you see, what is the MA? What is the sigma? What is the, for example, MC? What is them? How many nucleotides we have? 4. But here we have a sigma, we have a inosine, we have a methylguanine, methylcytosine, we have a adenosine, we have a lot of fucking material. The different nucleotide. Why? They don't want to translate. Just they want to be much more bigger, have one more methyl group, and bonds to whom? The tRNA should stick to which particle? Ribosome. They can easily bond to ribosome. You see exactly on the arm we have a different nucleotide. Do you see?
Yeah.
And you should know we have a 2 structure, secondary, tertiary structure. This is a clover leaf, and this is a L-type. L-type clover leaf. You should understand the chemical modification is methylated, dehydrouridine, zedouridine.
Okay.
What?
They have different binding sites, right?
Yes. The D loop is bonds to amino acid tRNA synthase. The anticodon bonds to codon. The T sigma or the other hand, other arm, it bonds to ribosome. And we have also 3 prime. It's a clover leaf model. 1, 2, 3, 4 is very important. Synthesis of amino acid tRNA. First of all, amino acid says, why should I come to you? Why should I come here? I need energy. Okay, I give you the energy. I produce amino acid adenosine monophosphate plus tRNA, and amino acid tRNA is produced plus adenosine monophosphate.
Do you have to remember this one?
What?
Do you have to remember this one?
Yes. Not the structure, but you should know the reaction for sure.
Really?
It means if you remember amino acid, you see the amine, carboxyl, carbon.
Yeah.
Plus ATP. Amino acid AMP plus pyrophosphate is produced. That's very important. Then amino acid AMP plus tRNA, amino acid tRNA plus AMP. It means AMP go away.
Yeah. Because
for sure you should know this alphabetic. If you don't know, you fail. For sure. If you know the structure, it gets more point.
Okay. So you have basically amino acid and ATP, and then it takes one of the phosphorus, right? Binds to the amino acid.
Yes.
Yeah. Left with 2
other phosphate go away.
Yes.
And finally, the carboxyl, instead of OH, it comes to the OH of this. And you see from here, exactly from.
What happens after this one?
This phosphate go away. See, this is between 2 O. Phosphate is between 2 O. Now, between 2 O, instead of phosphate is tRNA.
But after this part, this is the same, right?
Yes.
And then this part goes with tRNA.
Yes, this part is gone.
This part, this goes away.
This goes away.
Uh-huh.
And who remain?
This goes away. Okay.
tRNA.
Uh-huh. and this O goes together with this one.
One of my friends asked me, Mahsa, this is like that, and said yes, but actually it's better. It's written here tRNA with AMP, because already the tRNA have here AMP.
Yes.
But this AMP go away. The adenosine monophosphate with amino acid go away.
Okay, but then this OH goes away, right?
Yes, OH with this H produce water. You see?
Oh,
you can write here.
okay. True.
Just remember the word.
Okay.
Best.
Okay.
The prokaryotic mRNA, eukaryotic 70, 80, these are very, very important.
Yeah.
Don't forget.
Yeah, I know this one.
Method of study translation is an in vitro technique. In vitro means in laboratory. It's cell-free detection by such a fucking method, but you should remember. And this one. What is that? Direction of centrifugation and UV absorption. Source density, sucrose density gradient sedimentation of a polysome. What is the polysome?
It's ribosome, isn't it?
A lot of
Ribosomes
ribosomes
in a chain.
during transcription,
Transcription.
translation. During translation, it is the mRNA, and a lot of
Yeah.
ribosome do the translation. Yes.
Yeah.
For example, this shorter, and this is a Christmas tree or
Christmas
feeder
tree,
or
yeah.
feeder, and we do a monomeric ribosome of fucking
But the Christmas tree, is it only in prokaryotes? Or both?
both
Both? Okay.
polysome. But this is much more observable in prokaryote because they do it exactly, I don't know, somewhere, but they we have a bond ribosome, we have a free ribosome. Extent is different, but in the prokaryote, in the bacteria, it's observable more.
Okay.
If you are confused on the exam, it was in prokaryote or it was in eukaryote, please don't say anything. Just say, And when the teacher asks it, is it in prokaryote or eukaryote?
Yeah.
A little bit think and said eukaryote. Why? Because most of the interesting thing happens on the eukaryote. Good?
Okay, good.
Do you learn it?
Yes.
And when the teacher said, what do you think? Is it good or bad? You should say, it depends.
And then it's like, what depends?
It depends, because always everything is good,
Yeah.
but maybe bad. This drug is good, maybe bad. Who knows?
Yeah.
It depends.
Okay.
Now we stop it and then we continue again.